Good morning to those who are in the morning. Good evening to those who are further east, Japan, Australia, New Zealand. Welcome to this Target 2035 webinar in association with the Chemical Probes Portal on improving best practice selection and use of chemical probes. My name is Professor Paul Workman. I am a professor at the Institute of Cancer Research doing work on drug discovery and chemical probes. I'm also the director of the Chemical Probes Portal. And I would like to welcome my co-speakers. This is the plan for the session. I will give a brief introduction, setting out the background to chemical probes, and then introducing Lenka. The main purpose of today is to hear from Lenka about her exciting new research just published on the currently rather woeful degree of compliance with best practice for chemical probes, and then we'll move into a discussion. And in that discussion, we will have, of course, Lenka and myself will be joined by Guillaume Lessene, who's a professor of medicinal chemistry involved in drug discovery and chemical probes research at the WEHI Institute in Melbourne, and Felipe Ossa, who's one of the editors at Nature Communications, who've been, I must say, have been very good at working with the chemical probes community to help us to get the messages out to the community about best practice. So this is just my introduction slide. Its title is Revisiting the Promise and Perils of Chemical Probes, Progress and Challenges 8 years out. And the reason I mention 8 years out is next month it will be 8 years from the publication of the Arrowsmith et al. Paper, where the SGC and a group of us in the community came together with a call to action for improving the research quality and best practice and the number of chemical probes available to the community, and to announce the formation, in fact, of the Chemical Probes Portal itself. And I just wanted to show you a picture of our wonderful Drug Discovery Institute at the ICR, which is where we discover our drugs and our chemical probes. So the context is that there is, as many of you on this webinar will know, but there may be some new joiners or some new people to the field. So I'll try to give the background as well in this presentation. In some ways, the community was alerted to difficulties with research reproducibility, and I think what's even more important is the robustness of research by the Bayer project, which found, and also the Amgen experience, which is summarized here. And these slides will be available, by the way, to anyone who wants them. And this webinar is being recorded, so it will be available for you to watch again, or for others to, particularly in the US and North America, to join us. So there was the Bayer and the Amgen projects that found a rather low level of ability to reproduce findings, and Monya Baker's article about the reproducibility crisis in Nature. And also in Nature was a separate, very nice review by Jonathan Beale and Michael Walters, who introduced some of the worst offenders in the use of chemical probes. Hopefully most of the compounds we'll talk about today will be much higher quality, but there are two problems in the field. The worst offenders, which are the compounds that interfere with assays or act promiscuously across multiple molecular targets, and they should never be used in chemical probes research. And then there's the other compounds that simply don't have sufficient selectivity, and one has to be aware of how to deal with, recognizing the fact that no chemical probe is uniquely selective for any particular molecular target. I think one should start from that position. So what do we mean then for the new people who've joined us? What do we mean by chemical probes? This is what Wikipedia tells us, and it's actually not too bad. It tells us not to be confused with hybridization probes, molecular probes, not to be confused either with imaging probes. We're talking about, in the field of chemical biology, a small molecule used to study or manipulate a biological system, such as a cell or an organism, in such a way as binding to the particular protein target of interest, and in that way changes the phenotype of the cell, and through that, the chemical probe can be used to determine the function of the protein with which the probe interacts, and it can also be used for validating molecular targets in drug discovery. Very nicely, from the portal's point of view, the Wikipedia gives the link to the chemical probes portal as the main link for where to find information on this, and the two reference. And oh, by the way, when you look on the portal, the definition is very similar to this, but we make the point that chemical probes are usually inhibitors. Again, that's for those who may be new to this field. Sometimes when we say to biological scientists, what we're meaning is chemical inhibitors as a type of chemical probe, it becomes more familiar. And these are the references that to the Arrowsmith article and to Derek Lowe's blog, and many more references available on the Chemical Probes Portal Information Center. So why do we need high quality chemical probes? Because the literature really is polluted with poor quality compounds with low selectivity, poorly characterized, unconfirmed mechanism of action, poor physicochemical properties. And what we see is that even after publication, and many publications from the chemical biology community, the biomedical research community continues to use low quality chemical probes. Today's seminar by Lenka is more about, once you have a high quality chemical probe, what's the best practice for using that? But we need to be aware that behind this, and Guillaume will probably comment on this, is the need for producing the high quality chemical probes in the first place. And just quick examples: staurosporin, multi-hitter kinase inhibitor inaparib, PARP inhibitor that never even went into clinical trial, doesn't hit the target, should never be used as a chemical probe. And then we have something like this P38 inhibitor, which you can see inhibits a bunch of other kinases. It has a low selectivity index, even though it's potent for the target. And there are thousands of publications, and most of these use old, out-of-date inhibitors. So this is characterizing why we need chemical probes in the field, and recognizing that as I said in the introduction, the community came together, really catalyzed by Structural Genomic Consortium, SGC, who have just been wonderful in activating the community and organizing these webinars. As you can see, I was a co-author on this, and many in the field were, alongside Cheryl and Arlette Edwards and others, and Suzanne Muller-Knapp. We said in the abstract, and I remember drafting this for Arlette actually, chemical probes are powerful reagents with increasing impacts in biomedical research. This is 2015, remember. However, probes of poor quality or incorrectly used give misleading results. And to help address these shortcomings, we'll create a resource to improve the quality and convey a current best practice. And Lenka has used that resource to select the chemical probes. Excuse me 1 second. Still drinking my early morning tea. So Lenka has used our resource to select the chemical probes that she used for her fantastic analysis. So I wanted to show you how the portal works, and upon which Lenka the basis for her selection of compounds. So Chemical Probes Portal is a website. It's an online resource to help scientists find and use high quality chemical probes, particularly to help biologists who are unfamiliar with the chemistry. It's aimed to make it easy for non-experts to select the right chemical probes before they start study, and to help them use the probes once they've started to design an experiment. We've already covered this. It was established July 2015. It's been cited almost 750 times. It relies on expert recommendations. Big thank you to all the experts who do the reviews for us. There are about 200 of them. We call them the SERPs, the Scientific Expert Review Panel. It provides an alternative to reliance on citation rates, Google, Wikipedia, or vendor catalogs, which are all flawed and usually historically and commercially biased. It provides an expert review mechanism for large-scale, to complement large-scale resources such as ProbeMiner and drugs and probes, and it has a host of useful information. That's what if you go to www.chemicalprobes.org, that's the website that you see. You can see it's a Google type search engine that you put your target into or your probe into, and you'll find information on it. This is the team that runs the portal. I'd like to thank my senior colleagues who are at the top and the staff of the portal who've been hugely motivated and hardworking in helping to move the field forward, working with the Target 35 group. This is our fantastic board of governors chaired by Milka Kostic, who I know is going to run a workshop in her institution using the recording of this as a basis. Thank you, Milka. And thank you to our board of governors and the US board that runs the residual activity when the portal was initially initiated in the US and now has a residual presence there mainly for administrative reasons. And thanks to them for their support as well. So a huge number of people, and most importantly, the SERPs, the 200 expert reviewers who carry out our reviews. So how does the portal work? Basically, a user or alternatively the curator, Dom, will suggest a compound to be reviewed. Maybe they've seen it on SGC, maybe they've seen it on donated probes or OpenMe, maybe they've seen it in a publication. And through the submission wizard, which is very easy to use, the compound is recommended, and then Suzanne Muller-Knapp and Dom, the curator, will work together to get 3 reviews from our expert panel. The reviewers will give a star rating to the review, and then the reviews appear on the website with the star rating. You can see 4 down, 4 is the top quality, 3 is the next best, 3 and 4 are highly recommended as chemical probes, 1 and 2 are less, so indeed 1 is not recommended. Then these will then sit on the portal and be findable by the user. So what are the criteria that we use for, that reviewers use for selecting chemical probes? These are well established, really over 10 to 15 years now, and I have to say are still not consistently applied in the community, which is a disappointment. But the increasing use of these criteria, I think, is good to see. Biochemical potency, you can see less than 100 nanomolar, cellular potency less than a micromolar. Wider profiling across the proteome is desirable, but selectivity at least 30-fold within the family or to the nearest target. If it's outside the family, is crucial to get a high degree of, acceptable degree of selectivity. And biochemical proof of target engagement, which Lenka hasn't looked at in this review. I think that should be the subject probably of the next study. And then there are desirable properties to have as well. I think perhaps we shouldn't say pains behavior anymore. We should say no frequent hitter or nuisance behavior from the compound. An inactive control should be available. The data, of course, should be available publicly, must be available. A second orthogonal probe is desirable. Aqueous solubility and stability in plasma is important as well. The metrics you can find, actually this is updated from our recent nucleic acids research paper here, which is the most recent review in the NAR database, most recent review of chemical probes portal, and the progress that we've made to build. When the portal started out, there was a small handful, literally, I think less than 10 probes on the portal when we started out. We have more than 900 compounds on the portal, of which 253, I should say, are historical. We now call them unsuitable, which are either superseded or are members of those horrible frequent hitter assay interference compounds that I showed you a cartoon of earlier. And there, it's important they're on the portal because in that way people can find the undesirable compounds or check if a compound they want to use is in fact a member of this group that should really never be used. Nearly 650 higher quality chemical probes, of which nearly 400 are 3 to 4 star compounds, more than 200 reviewers, 335 targets have 3 to 4 star compounds, 1,000 reviews overall on the portal. So a rich source of information for the biologist and the biomedical researcher, as well as the expert as well. And you can see we're diverse, we're covering multiple countries, and really the activity has been growing over time. In terms of using the portal, a user would come in onto the website, would browse, You can browse, you can download, you can go to the information center to get information. You can type in your target of interest. You'll get the probe results with the 3 or 4-star or other recommendations. It'll give you information, concentration, potency, selectivity, and that will allow the user to make an informed decision on whether to use that compound or go to others, which will also be flagged up. In addition, if the target isn't covered, because we don't have full coverage, in addition, you can go to the CANSAR resource, the probe miner resource, or the probes and drug resource, and others as well. This is what the website will look like. When you click on an approved probe, you'll get 2 SERP comments, you'll get the star review, and you'll see the recommended concentration to use. Here we have a review recommending use in cells. Here we have a review recommending use in rodents as well. This is an example of a superseded unsuitable compound, an old PI3 kinase inhibitor that's still very highly used, hundreds of reviews each year. It's been discredited and not recommended for more than 10 to 15 years now. These are the types of compounds you should be using. You will get a flag for a compound that's not recommended by the portal with now unsuitable compound, and you can get this other information as well. We're also beginning now to introduce criteria for new types of probes for PROTACs, for molecular glues, and for other new classes of compound. And you can also, so that's in the information center at this link. It's easy to find. And you can also find the extra criteria that are required if you want to put the probe into, for example, a mouse model. And there's the link for that, which again is easy to find. Beyond evaluating probes, this is the range of information available in the information center and elsewhere on the portal. There's tutorials, frequently asked questions, presentations, links to key publications, resources, the unsuitable compounds, and the like. You can browse, you can download, and you can spend time and learn more about using probes. So just before I introduce Lenka, I just wanted to tell you about where we're going next. We're aiming to significantly increase the number of compounds in the next 5 years, of course, working in close partnership with the Target 2035 movement, which we're closely involved with. We want to increase the number of compounds, increase the diversity of probe types, more glues, more probes, PROTACs, and so on, increase the number of and diversity of targets, and the number of protein families covered. We launched the new website in 2022. We hope you like it. It's easier to use. It's full of information. It's got much more content and many more probes listed. It has an improved search and database structure. It's easier to submit a probe. Please submit probes to us. Please help us to review and join the SERP panel as well to help us do that. Outreach is super important if we want to change behavior in the biomedical community. We need to create a community of users, a wider community of users out with preaching to the converted or preaching to the choir. We need to preach to the whole biomedical research community and to engage them through conferences, webinars, hackathons, social media, through training and that we provide on the portal, and you can find it elsewhere with links that we provide as well, and especially reaching out to the biology users and their organizations too, to alert people that there is a problem and that we have solutions to that problem. So these are the different organizations that we need to engage to work with to change best practice in the community, and well represented, I think, by the panel. Just rehearses the criteria once again that Lenka used for her study. This is to acknowledge the super team that I work with, and especially to acknowledge the expert review panel. Please join us. Please contact us. There's a contact us button on the portal website. And now this is to introduce Lenka, who's professor of pharmacology and related disciplines at the University of Sydney. She's produced this wonderful paper that was just a study of use of chemical probes, just published over the last weekend. This is her title. You will see that Lenka is wearing a red coat here. Unfortunately, she's not wearing red today, Lenka. I'm disappointed that you're not wearing your Man United colors, but it's just discovered with Lenka. Lenka and I share a love for the soccer team Man United. So apologies to those who support other teams. And this is Lenka's paper that she's going to take you through. So with that, I'm going to stop sharing my screen and hand over to Lenka to give her presentation. Thank you.
Thank you, Paul. So I put my screen up. While I'm doing that, I just want to disclose that I was a Man United until I got married to a Liverpool fan. So I have to be very careful with Manchester United, but surely, yeah. So can everybody see my screen?
So Lenka, this is now the presenter's mode and not the full screen mode. There we go.
So now, all right. So it's
Yeah.
just I will be looking away from the camera then. That's okay. All right. So thanks, Paul. Thanks, everyone, and the opportunity to talk to you about this systematic review that we have just published in Nature.com. So I would like to give you a little bit of background before I talk about the data, how we got here, because I'm not a qualitative scientist. I'm a medicinal chemist turned pharmacologist. And my team works, my lab is focused on glioblastoma. So we are studying signaling pathways in glioblastoma cells in the quest to find new targets, new drugs. And more recently, we have also become interested into the drug-tolerant persisters. And many years back, that was around the time when the portal was launched, I also stumbled across this kinase inhibitor that was developed by Boehringer Ingelheim, and we observed extraordinary efficacy of this compound in glioblastoma cells, whereas all the other inhibitors of p38 or MK2 did not. So after a few years of trying to understand what was going on, we identified that this is actually a tubulin inhibitor. It inhibits tubulin polymerization. And this is how I've learned that molecules inhibitors have a night life. CMPD1 proved to be a good lead for the development of brain-permeable chemotherapy for glioblastoma. So that is part of the research that we are still doing, going through the preclinical trials. But at that time, I also published a paper on the non-kinase targets of protein kinase inhibitors. This was in 2017. When Paul published the famous Cancer Cell Review, and where he and we both provided these guidelines about how to use chemical probes, how to improve the robustness of biomedical research. And then I was following the community, how the probes were emerging, and I was quite disappointed noticing that the guidelines, although people started using these chemical probes, the way they were using them were not ideal. And then the virus came, and what happened at the same time was that this was a coincidence, and it was a little bit of a storm for us because Sydney Uni started a new curriculum that was coming into the students' life in 2020, when we suddenly all went teaching and lecturing and doing research on Zoom. And in this year, I was also charged with the curriculum development for our honours students, which in Australian system is the 4th year undergrad students, which is the stepping stone for them to come to do a PhD. So giving all these things, I thought, why don't I teach them how to use probes, what the probes are, and how to use them. So together with Jennifer Byrne, who is quite famous in the space of the primers and the molecular probes, we developed this module. And part of the assignment for the student was to select a probe. So we work with the chemical probes portal, because I think that's the most user-friendly portal, especially for non-chemists. Students select any probe and then they do work around it. And part of the assignment was to find 3 publications and address whether the probe was used correctly or not. And when I was marking those assignments, I could see that on average, 2 out of 3 publications that students reviewed were using the probe incorrectly. So the maths told me that 66% of research is still done incorrectly. And because we were for 2 years in a lockdown down here in Australia, out of the lab for most of the 2020 and 21, I took my PhD student, Jaden, so he's here, Jaden, and I said, why don't we just look at this in more details and try to quantify? We worked closely with our chemistry collaborators, Jen and Adam at the University of Newcastle. So this is all my co-authors on the paper. and they've been great for all aspects. So we went through 5 rejections, but then we found a good home, and the paper just appeared online over this weekend. So what have we done in the paper? I have selected 8 probes. There was no bias. These probes were randomly selected. But what I was looking at is to look at the probes that were covering fields of different sizes that were older. So I really wanted to look at the probes that have been in circulation for a while, probes that have these inactive, matched inactive controls. Ideally, that would be good if there was also an orthogonal probe available with the selected probe. I also looked at the number of publications because this had to be done all manually. And then we were trying to pick probes that were having high writing either on the probes portal or the probe miner. So this is the 4 epigenetic probes that we have selected targeting EZH2, KDM6, CREB-BP, and G98. And then we also looked at the use of 4 kinase targeting probes. So 2 were targeting Aurora kinases. Then we had an mTOR probe and a CDK7 targeting probes. What we did next was we tried to find publications, and this kind of turned out to be a huge project. So we did 3 searches just to try to get all the papers using these probes. We used PubMed, where we searched by the keyword. We also searched in SciFinder by the keywords, and we did the searches by the structure. This has given us the nearly 1,200 pages of references. This was just the titles and the authors, not abstract. It was a massive document that had to be then curated by Jaden and Jen. Patents were removed automatically, but the replicates had to be removed manually. We ended up with over 1,000 publications. Could not retrieve a few that were not written in English, or we did not have a full text access. And then we've got these more than 1,100 articles that we then reviewed for eligibility criteria. And what we were looking for, the eligibility criteria, there was only one figure using the given chemical probe in a cell-based assay. So we have excluded medchem studies. This is the typical hit-to-lead optimization where the probe was maybe used just for the benchmarking. We excluded papers that had no mechanistic experiments, clinical trials, in vivo studies. We were purely looking at the cell-based assays, and then all the reviews and editorials and the comments were excluded. So at the end, we came to 662 publications. And in these publications, we had at least one figure with the given probe used in a cell-based assay. We were still quite selective, and few assays were not taken into consideration because there you have to use a higher concentration. For example, when you're doing those response curves, or when you're looking at CETSA, the target engagement inside the cell, or a toxicology assessment. So what we really analyzed in these 600 papers was mechanistic target validation studies and signaling pathways. So the first thing that I was quite surprised to find out once we got all these papers was that only a fraction of research is done with the highest quality probes. So when you look at the size of the field and then the number of the papers, and this is before we excluded all the reviews or commentaries, the percentage, maybe exception is THZ1, the CDK7 field, but the percentage is quite low. So I was surprised that how else is KDM6 field studying this lysine demethylase, because GSK-J4 is the only probe that we currently have. However, only 12% of these papers was using a chemical probe. But going back to our paper, and what we tried to look at was just 3 basic questions. And I know Paul mentioned we didn't look at target engagement, and it was in our to-do list at the beginning, but it's just turned out to be extremely complex and and very frustrating because everybody had a different model. So the target and engagement was quite difficult to assess, especially in the papers that were not within my expertise. So what we did is we looked only at these 3 questions, and we were analyzing whether the chemical probe has been used within the recommended concentration, if the inactive control exists, whether it has been employed, and if there are orthogonal inhibitors in that space, whether this has been employed as well. And then I show you on the example of UNC 1999, which is the probe targeting the methyltransferase EZH2, how we assessed each paper. So in regard to the concentrations, firstly, we looked what's there online, what is the portal, or we also looked at the Structural Genomics Consortium website because that also has information. So we looked at what concentrations are recommended by these 2 open access portals. And then we looked what people did in their papers. So if all experiments were performed below this recommended maximum, we call the paper complying. Now, it was quite difficult to assess these concentrations because lots of papers would have some assays performed with concentrations that was okay, but they would still have some assays with a much higher concentration. But just to be fair to everybody, we said this would be a partially complying paper. And the publications that had the probe always at these high concentrations above the recommended maximum, we call them non-complying. When we came to assess the inactive controls, there was only 2 answers, yes and no. The probe, the inactive control molecule was included at least in 1 experiment. And we were quite nice to everybody. Some papers were only mentioning that they have done it, but data not shown. But we still consider this paper as a yes, complying. And if the paper was completely done without the inactive control, we called it non-complying. And the same when it came to orthogonal inhibitors. If any orthogonal inhibitor was used, it doesn't have to be a probe, and we did not look at the concentration of these orthogonal inhibitors. We said, yes, this paper complies. If the whole study was performed just with UNC 1991, 1 probe, in that case, we said no, and it's not complying. And so we assessed all these papers for these 3 questions, and then we graphed it. So here are the results for UNC 1993. So you see approximately 1/3 was always below the recommended maximum. 1/3 of the studies was in that gray area, yes and no. 35% was using much higher concentrations that is recommended. 88% did not include the inactive control UNC 2400, and approximately half would confirm their data with another EZH2 inhibitor. So we have done this for all 8 probes across 662 papers, and this is the summary per probe, where the orange means non-complying, blue means complying, and gray is the gray area in between. There was a variability quite different. So for example, THZ1 was always, it was always used at the right concentration. I think we found only 2 out of 140 papers that used THZ at the higher concentration. But the UNC 0638, that was, for example, used largely over the recommended concentrations. When it comes to the orthogonal inhibitors, it was usually half-half. Half of the paper did use more than 1 probe, half did the whole study with 1 probe. And inactive controls, that was the biggest surprise. 90% of studies were completed without using the inactive probes. So when it's all pulled together, these are the values. Average of these will come down to 66. What I saw when I did those assessments of the students, and I saw this is quite bad. 2/3 of research is not done properly because the probes are used incorrectly. We also looked at the number of citations, and this was maybe more worrying than the use of the probes itself, because these 662 papers received up to 15,000 citations. And the percentage of citations was also largely reflecting the compliance with these guidelines. So those 92 papers that, for example, did not use the inactive probe, they were receiving the majority of the citations. So when I saw this, what obviously came to my mind was that biomedical research is the blind leading the blind. There is a incorrect use, and people are unknowingly producing more data following these incomplete experimental designs. So these numbers are not very promising, and I think they are even worse in the reality, because I would like to point out that these numbers are based on the benefit of doubt. So we were really very nice, and the concentrations, even the partially complying papers, are counted in this 78%. So I personally don't think that 80% of papers is done with the right concentrations of these probes. Inactives, very often it was not shown, but we still counted this as complying. And when we looked at the orthogonal probes, again, we did not look whether it is an established, approved probe and what is the concentration. So these data, I think, are better than the reality. And they are only assessing compliance with one guideline. But as we know, we need to comply with all three to really get a robust data. And so in our cohort, what we looked next was that how many papers did comply with all three guidelines. So 267 publications related to these probes should have complied with all three recommendations, because these chemical probes come with an inactive control compound, and there are also orthogonal inhibitors or chemical probes in the space. And unfortunately, what we found was that out of these 267 publications, only 11 publications, which is 4%, did comply with all three guidelines, and the rest did not. And that was disappointing. And I think looking at these papers for nearly 2 years in and out, I think this is the sad reality that we are facing in terms of the quality of biomedical research. So before I talk briefly about how to move forward, I just would like to highlight the winners. So these are the 11 papers that did the right thing. And the one thing that really popped out immediately was that on these 11 papers that were beautiful, brilliant in terms of experimental design using chemical probes, there was a chemical biologist as a co-author. So that kind of tells me that the chemical biology community knows what to do. We have to preach much more than we have been doing so far. So in addition to this observation, this is just what we have observed, was that the small proportion of studies was done with these high quality probes. We still found these historic or pain compounds in circulation. Many studies, what is happening often is that many studies first determine IC50 values in a viability assay. Unfortunately, the GR metrics that are developed by Peter Sorger, that platform is not really used at all, and it's a shame because it's a much more sensitive way of determining drug sensitivity. So people normally do this viability assay, determine the IC50 value, and then they use that concentration in their mechanistic studies. As I said, well, Paul asked about the target engagement. Yes, we wanted, but it would be even more frustrating that what we have reviewed, because we did not see in the vast majority of papers any evidence of target engagement and modulation in the cells. And orthogonal inhibitors, if they were used, they were very often outdated historic compounds used at high concentrations and not always orthogonal. I think that concept of what is an orthogonal inhibitor is not that well engraved in the biology community. So overall, My feeling is that lots of this biomedical research data is lacking robustness. It just follows this outline here. There is a rationale. Target X is important in disease Y. One takes a molecule, puts it on the cells, the expected phenotype is observed, and there is a conclusion that targeting X cures disease Y. And so this brings me to the problem of robustness. So I personally see biomedical research or drug discovery as a house of cards. And we know that the success of this house depends on the target identification and validation. And target identification and validation strongly depends on chemical probes alongside other cards. So 10 years ago, and Paul mentioned this, we were talking a lot about the reproducibility. I don't want to say that these studies we reviewed are not reproducible. I think anybody using one of these probes at the 50 micromole would reproduce the data of the paper. What I think is that these studies are not robust, and it's because basically they're missing the chemical probes card. And if you played House of Cards with your kids, you know when you remove the card, what would happen. So how can we improve this? I think the moving forward is to educate especially the biologists. And when my colleagues are asking me, why do I need more than one chemical probe? I always say, look, any molecule will hit 40 to 400 targets. I have evidence, this paper that I always cite, and even the most selective inhibitors, they will have a night life. So you can never assume that if you take even the best chemical probe and you put it on the cell, that the phenotype is purely related to inhibiting the primary target. So I always say this example, you need to take the inactive control, which is your scramble RNA, if you are doing this with molecular knockdown. And then I say, and if you're doing molecular probes, you would always take 2 or 3 SG or SH RNAs with the different sequences, just to be confident that the phenotype you're seeing is related to your target. So the same concept goes for chemical probes. You take, for example, UNC 1999, and then you need to take something that looks differently. So this is where we also put in our review all these structures, and the ones that were within the same family, the pharmacophore were the same. We were putting them into these frames, because I noticed with biologists, they don't really understand when I tell them you need an orthogonal control, what it really means. So I said, just look at the structure and it has to look like apples and oranges, so slightly different. How this is a timeline or a flow chart we were asked to put by one of our reviewers, which was a very good recommendation. I was guided here by Paul's review from 2 years ago in the Future Med Chem. We just modified it slightly, but this is, I think, a very simple flowchart that would, I believe, could help all the biologists to find the right chemical probe. So I think what we need to do is to tell the biologists about it and guide them to the right presentations, papers, or just guide them how to select and use these probes. So in our paper, as well as in Paul's paper, there is also all these seminars, either from the Target 2035 or the seminars that were given in the past, and as well as this seminar about the chemical probes. I think if every chemical biologist will have one slide on chemical probes in every talk we give now, especially when we are back to conferences, I really hope that these grim statistics that we have just published will be slightly better. What was also asked by one of the reviewer, which is a great suggestion, and I'm grateful that we were asked to create a checklist for editors and assessors of the papers, how to go about assessing papers and then be guided what to look for. So I developed what I call a rule of 2. where I would like to see that every paper, every study that uses chemical probes will have at least 2 of them. So this could be either 2 orthogonal probes or a probe and it matched inactive compound. I think it is important to always preach the message that these probes have to be used at the right concentration, and there needs to be an evidence of target engagement of modulation. So as an assessor or editor, I would suggest that there is this checklist of 5 items. And basically what we need always is 3 yeses for the questions number 1, 2, 3. So use the right probe, use it at the right concentrations, and show in your disease relevant model that the target is really engaged and modulated. And then what we need is at least 1 yes for these questions 4 and 5. That's the another probe, either a inactive control compound or an orthogonal inhibitor. Sometimes the probe they cannot be both completed. That's why I said just have 1 yes to 4 or 5, and then I would be happy to approve this experimental design. If this is not satisfied, I strongly recommend that the paper or the grant, the feedback is given back to the biologist that you need to confirm your data with additional compound or using the right concentration. So in summary, what I believe and what we are saying in our paper is that 96% of publications, they use present suboptimal use of chemical probes. This obviously has serious consequences for biomedical research in terms of a bad education, wasted resources, time, money, and the drug discovery is obviously based on those house of cards when one of the cards is missing. So we are up, I think, for a culture change. And if you know, there is this culture change pyramid where we have to provide the infrastructure. And I think that's been happening given all this great effort of the Target 2035 Structural Genomics Consortium and all these initiatives around the globe to develop probes against as many proteins as we can. I think we are also in a good space of making this easy. I honestly cannot imagine anything more user-friendly than the chemical probes portal. There's nothing easier than that. And all these other websites, the Structural Genomics Consortium is also very easy to navigate and find what you need. What needs to improve is to make the inactive controls easily available. So the bad statistics is, I think, partially compounded by the fact that some of these inactive controls, they are available, but upon request, custom synthesis, they cannot be easily just click, order, and pay. However, GSK J5 and UNC 2400, they are both commercially available and they still haven't been used. As the scientists, as a community, we have to make this normative. So we all have to do it, which I don't think is the problem, given the 11 winners, they all had chemical biologists as co-authors, but we have to spread these words along, outside to the community. I strongly believe that only grants that have a robust experimental design should be funded. This should be something that, as a reviewer of the grants, we should be looking at. And then I believe or would like to propose that the journals are the gatekeepers and they really request that this rule of 2 or at least 2 chemical probes used correctly is a requirement for a submission or for being accepted. So that's all from me. I leave it here and I'm looking forward to the questions and discussion. How can we make this work better?
Lenka, thank you. Lenka, thank you. That was absolutely wonderful. I'd like to congratulate you and Jaden and Jennifer and Adam, who I haven't met. I hope I will at some stage on this truly important study. Previously, we were all working with anecdotes, the sort of anecdotes I showed you on staurosporin and LY compound PI3 kinase and the low quality so-called PARP inhibitor. Now we have, at least for 8 probes, very, very systematic analysis, and I think it's you should be congratulated on behalf of the community for this really, really important study. I'm sure it'll be highly cited. And the key thing, just before I bring Guillaume and Felipe into the discussion, and by the way, I will then be turning to the audience for questions as well. We have at least 12 minutes left. I think a key point that we need to get over is how can we preach beyond the choir? How do we preach to the entire congregation, or even the people that don't go to church, right? If biologists don't know what best practice is, or are unaware of this huge literature, and don't have a friendly chemist or chemical biologist, they don't even know necessarily that they need an orthogonal control or a second active control until Felipe, reviewer 3 at Nature Communications, comes in and says, this is not acceptable in order for this to be robust. But then, of course, reviewer 3 has to be contacted by the journal. The journal has to know that a chemical biologist should be amongst the reviewers. Anyway, those are just some initial comments. So I'd like to start off just by asking one question each to Guillaume and Felipe, and also give you an opportunity, by the way, the two of you to ask Lenka. But Guillaume, how do you think medicinal chemists can help biologists with this problem? How can we raise awareness? And in your perspective as a medicinal chemist and a probe developer yourself, how do you see things?
Yes, thanks. And thanks, Lenka. That was brilliant and much needed addition to the field and to resolve this problem. So I think the way I see it, at least here at our institute, is to try to break the barriers between biologists and medicinal chemists and chemical biologists, and to really have this dialogue and as much as possible to really have or have this understanding of what chemical probes are and how they function, and therefore that puts in a little bit more in perspective these issues, which I think, as you've highlighted, Lenka, the issue of concentration of use of the compounds is one of the ones that I find the most problematic in publications, and especially in the cancer field, because there will be a concentration at which compounds are going to kill cells. There will be at a toxic concentration, and I've worked a lot with colleagues in the area of the BCL2 and apoptosis, and that has plagued the literature for many, many years, where compounds had a dubious binding affinity against one of the pro-survival proteins, and then cells were treated at very high concentration, and it actually induced apoptosis, but there was absolutely no validation of the cause and the consequence. And I think trying to also make our biologist colleague to understand that treating a cell and seeing a phenotype and having some binding is not sufficient to provide evidence of mode of action. So the other thing that I wanted to mention, as you pointed out, is we're also developing probes, and that's almost an art in itself, because developing a drug has its own challenges. But if we think about developing a probe, selectivity is such an important factor to be able to say this is a good probe for target X, and to be able to demonstrate this very robustly is very important when one develops a compound. And I think one of the things that's highlighted by the probes you mentioned, Lenka, is that there is this perhaps this confusion between a drug against a target that has gone into the clinic. And then the assumption is this is going to be a good probe. It's gone into the clinic. It's a inhibitor of protein X and gone into the clinic, and there's this assumption, and forgetting, as you pointed out, that there may be other targets that are associated with the actual clinical activity. And there's this paper in Science Translational Medicine a while ago that showed that, in fact, the activity of these compounds in the clinic is actually due to the off-target effect, and not at all to the target they were developed against. So this is really important. And just to finish, I think the other thing that's really critical and is a bit scary as well is that as scientists have more and more access to screening facilities, and against their target of interest or even phenotypic screen, the ability to be going from the screen hits to a cellular assay is also something to stop as much as possible, because again, the activities that are usually the outcome of a screening exercise is nowhere near sufficient for to be used in a cellular model, and sometimes even they're used directly into an animal model, forgetting also about the MPK and all these aspects. So altogether, the development is also probe development and thinking is very, very important. And not forgetting that they are in some way drugs, but they have the challenges of needing to be very specific and with demonstration of specificity. So these are just a few points that I wanted to add to that, because, but I think it's a very important conversation to have.
Thank you, Guillaume. That was those comments were extremely helpful, coming from a distinguished medicinal chemist who works in this field. And I'm a biologist myself, although I studied chemistry to quite a late stage at university. I've always collaborated with chemists, and I've always collaborated with clinicians. And in multidisciplinary research, it's absolutely essential that you have colleagues who are expert in the areas that you are interacting with. If you're going to do a crystal structure, then you would talk to a structural biologist. It's not yet.
That's right.
It's not yet sort of common knowledge amongst biologists who are not used to working with chemists that there is a problem, and that 90% of the problem can be solved just by talking to a chemist who knows about these things.
Absolutely.
I just want to make I just want to make a couple of comments before I bring in Philippe. Laurent Gross in the chat asks about if an inactive compound is unavailable, and actually that's incredibly common. So there's even more of a problem, because very often there is no inactive control ever published, or if it's published, even it may not be sold, and if it's sold, it's not bought, right? So there's 3 levels of problem. And Laurent asks, is it useful to use a mutant allele of to put a mutant allele into the cell and ask if you get resistance? And I think that is a good biological control to do as well. But I would say ideally, and if you can't find other probes, then yes, please do that. If not, alternatively, do the experiment that was referred to by Guillaume, the Sheltzer lab experiment, which is to CRISPR out the target of interest and ask if the compounds lose or retain activity. If they retain activity, they're not working on the target that's claimed. And he found that for almost 20 drugs that were in the clinic. So we find that a useful experiment to do as well. And then Niaz Wali asks a question that we haven't mentioned: fluorescent probes. I did say at the beginning that the topic of this webinar is chemical probes, usually inhibitors, that are used for asking about the function of proteins. So we're not talking about fluorescent probes, for diagnostics or that type of analysis as well. So Philippe, I'd like to bring you in, and it's been mentioned a couple of times, the journals are the guardians or the gatekeepers of quality here. And I have to congratulate the Nature stable of journals for doing a lot. They published the conversation that Katarzyna, senior editor at your journal, had with Cheryl Arrowsmith and myself in an attempt to reach a broader community. They published Lenka's paper, despite the rejections that you heard from other journals. I think that was a fantastic thing to do. But more broadly, Philippe, how do you see it as an editor of a prestigious general journal that biologists will read, and how do you think the journals can help us more in this endeavor?
Yeah, so I think the first thing I can say is, well, the first is the disclaimer. Every journal, even within a family like Nature, each individual journal, as we like to say in our pro forma letters, is editorially independent, and that reflects on the fact that you have a vast number of different editors, and everybody approaches things slightly differently. But in my experience as an editor, you don't really want to get into technical arguments with people, because you want to stay away from telling people how they should do your research. So the idea you have as an editor is that you should reflect the best practices in the community, and part of that is done, and I think that's the key point, is by recruiting the correct reviewers. Obviously, the first step is awareness, and in that sense, you know, Nature Communications and the Nature family of journals are already taking steps. We have internal documents that we train editors on, which mention the problems that you have discussed about chemical probes and what to be aware of and what to look out for. But I think given the fact that we tend to try not to get into technical arguments, and that we want to really be a reflection of the community itself, the key stage at this point, from my point of view, would be to ensure that we have reviewers that follow these rules and that ask these things from scientists. Whether that is because, at least at Nature Communications, we often expect that when a paper goes out to review, there's some scope for improvement. So sometimes we will send something out in the hope that reviewers will suggest experiments that will make it better. This is not always the case. Some journals are stricter, and they very much want the peer review process to tick the checkbox of this is fine, and not to include more work. So this is a sort of philosophical difference. But what we look for is a reviewer that can also provide that guidance, whether it be you need to do these extra, you know, you are seeing something interesting, but you need to do these extra experiments to convince people and for the work to be rigorous, or whether it's simply giving us the tools to go back to the authors and say, look, an expert in this field has said that your data is not reproducible, therefore we cannot publish. So I think the reviewer finding is key at this stage, especially if you have already this population where, you know, 96% of papers aren't doing it properly. There will always be community pushback on the basis of, but you know, all these other publications didn't do it, or this idea of no one else in my department bothers to do this, so why are you asking me to do it? So I think that's and obviously, as I said, as editors, we don't want to get into these arguments with authors. So I think that at this stage, the reviewer finding is key. But obviously, these things evolve, and as things become best practices, I mean, if you go back in time, now in structural biology, it would be inconceivable that you publish a structure without a table that shows, you know, structure statistics. But at some point, this wasn't the way it was done, and you need a certain sort of critical mass. So I think from the perspective of what I can see, we're still at the stage of gathering that critical mass, and at that point, I think the best thing that we can do as journals, other than internally being aware of these issues, which we're obviously doing, is to really have these resources and push for reviewers to help provide us with this. And then obviously that dovetails onto the second point, which I think especially Nature has been good at with this, which is we want to provide a platform for the dissemination of this information. So things like publishing Lenka's manuscript, attending these webinars, commissioning appropriate sort of front half to promote these best practices is the other way of doing it. And certainly, you know, as this becomes best practice in the future, it may be more of a case of journals saying very specifically, if you don't have this, we're not interested.
Thank you, Felipe. That was extremely helpful and very, very good comments indeed. We're just 1 or 2 minutes over. Christina tells me that we can let this run for just 2 or 3 minutes longer. I recognize people will have to leave. I apologize if you have to leave. We will. I just want to continue for a couple more minutes for those who can able to stay on, and you can always watch the end on the recording. I just wanted to comment that I think I think your comments are absolutely right, Felipe, that it's not your journal's job or any other journal's job to tell the community what the standards are. It's to reflect the consensus views of the experts in the field. I think the problem that we have with this, in contrast to structural biology, it's a bit different in the following way. If someone submits a structure of a new protein, it will automatically be reviewed by 3 or more structural biologists. If a researcher, a biologist publishes a paper and the final panel and the final figure, to complement all the genetics, is to throw on an industrial dose of a chemical compound, and then it goes out to review biologists, it will not be picked up because the theme of the paper will not necessarily be seen as, chemical biology. So I think that's one of the cultural things that we have to overcome. And, we work very, very hard to put out these messages from the chemical biology community, now Lenka's contribution as well. If the general, and this is, by the way, this is not being critical of the biology community, it's trying to raise the awareness that we can help you. There are issues here. And I would just say one point, Felipe, that the problem is not necessarily reproducibility, it's robustness. So you can take a terrible compound, do the experiment 20 times and reproduce it every time. It's still wrong because it's the wrong reagent and the finding is not robust. And that's why you need, and I think the comments that if you have to push back, hopefully with the help of a reviewer, then the comments are around, you have not included the correct controls. And there are numerous papers and guidelines and so on that you can cite, and hopefully reviewers will help you to say, unless you use these extra controls, it's not a robust finding. So I just want to comment that Rahim Salim, in the final question in the chat, has asked, is there an active effort to discourage work on pains? Yes, there is. And by the way, I do think we should probably stop calling them pains. I think they are frequent hitters, nuisance compounds, promiscuous compounds, because pains is a slightly misleading, meaning assay interference, actually, which is one type of pain. So yes, we and others on the portal and elsewhere have been encouraging people to discourage work on pains. People continue to use staurosporin as a kinase inhibitor for reticular kinase. People will you can still buy inaparib, the PARP inhibitor that doesn't actually inhibit PARP and failed in a clinical trial. You can still buy that as a PARP inhibitor. You still using curcumin and sirtuins and so on. So it will still happen. We just have to continue to work hard. Preaching beyond the choir into the broader discipline is a difficult thing to do. Lenka's contribution, I think, has been massive to help with that. One thing that we are going to do, Felipe, at the portal, our next step is to produce a draft white paper that will summarize the best practice findings for developing and using a chemical probe. We'll then contact all the journals, contact everybody in the field and beyond if we can, heads of biology institutes and so on, and try to produce a consensus white paper document that the whole community can own. And then I think that might be a step towards the editors, Felipe, having something they can point to as a higher profile and more strongly a widely supported document. So I just want to turn to Felipe and Guillaume and even Lenka. If we're 6 minutes over now, but do you have any final, final comment that you would like to make before we close? And thank you again for joining. Lenka, anything you'd like to come back with?
I think I said it all in the paper, and I just would like to thank again Jaden, Jen, and Adam. It's been, it was a systematic review. We have never done that before. We were learning it as we were doing it, and we didn't know that we were doing it until the editor told us. So that was quite helpful. I just would like to stop seeing these great papers, but sometimes they are just badly done. It's a waste of money and time. If we can stop that, then I would be happy.
I just wanted to very quick, just to summarize one thing that you said is trust and consult your medicinal chemistry / chemical biologist colleagues.
Absolutely,
because
absolutely.
They'll be able to pick up things before a bad experiment is run. And I agree, it's not done on purpose. It's just also understanding the issues. And
absolutely. Felipe, final comment?
Yeah, I think what you were saying, Paul, is very valuable. I can certainly tell you that even if it were a tail end figure for structural biology with one small thing, we would always find a specialist reviewer for it. So maybe one of the things that we need to discuss and think more about is having this awareness of, okay, even if it's a small part, this is something important enough that you have to go and ask a medicinal chemist. But that also needs some cultural receptivity in the community, because obviously one of the things that can be difficult for us is getting technical reviewers, because people will read the abstract or the title of the paper and they'll say, this is nothing to do with me. I won't
Yeah.
review. So it's kind of this
Yeah.
symbiotic relationship, let's say, in between us and the community. But I think with the support that you're building, this is definitely a direction that we can go.
Thank you so much, and thank everyone again, and Christina and the Target 2035 and the portal team for organizing this. Lenka, your contribution and your colleagues, I think, will be a fantastic contribution to the field and will help change practice. And thanks to everyone for joining this. I hope you found it useful, and please join us again for future webinars. Please use the Chemical Probes portal and Target 2035 facilities. And thank you again.